Alrighty, so today we're back and we're doing another video. We are doing the pathogenesis of HIV infection. So what we're going to go through today is we're going to write out a list of steps, right, that define how HIV infection gets from our blood, how it affects our cells and establishes latency, right? To begin with, we're going to have a quick overview of what the HIV virus looks like, and then we're going to go into a bit more detail into each of the steps as to how it promotes infection. All right, so to begin, here I just have like a super rough diagram of the HIV virus, right? Okay, so what we have around the side is we have an envelope, okay? And this envelope is actually sourced from the host cell membrane, right? When we go into a bit more detail later, we're going to see that when our HIV virus buds off, it takes the envelope from the host membrane with it, okay? So here we have an envelope. And I've drawn two surface proteins, okay? There are more on the HIV virus, but two main ones that we're going to discuss, okay? We have one that's called the GP120 and another that's called GP41. Okay, inside we have a capsid, okay, also called a nucleocapsid. And this is almost like the house of the nucleic acid core, okay? And interestingly with HIV, it's cone-shaped, okay? And then finally in the middle we have our nucleic acid core. And you can see here that I've drawn two single strands, like two single-stranded RNA, two single-stranded RNA strands, okay? And within there we also have some enzymes, okay? Some enzymes of virulence that further facilitate HIV infection. But we're going to go into a lot more detail in a second. Okay, so now we know what the HIV virus, really roughly in a crude sense, what it looks like. We're going to talk about how it invades. Okay. So step one, adhesion. So what HIV does, right, is this glycoprotein here, GP120, it binds to the CD4+ receptor on a range of host cells. Most notably, you might think of the T helper cells possessing this glycoprotein, but as well we have macrophages, dendritic cells as well. Okay, mostly immune cells, the cluster of differentiation protein. However, interestingly, research shows that once GP120 binds to the CD4 receptor, HIV actually still can't propagate. It cannot infect another cell, which begs the question, what else is involved in HIV adhesion? And that's what we refer to as co-receptors come in, okay? I've written two main ones there, so we've got CXCR4 and CCR5, but there are a range of others, and what these are is these are co-receptors that GP120 also has to bind to facilitate fusion, which we're going to get to in a second. Using these two as an example, as the main ones, we see CXCR4 is predominantly involved in more advanced HIV disease, whereas CCR5 is more implicated in more newly onset HIV infection. All right, so once our GP120 has bound to the CD4 receptor and also formed connections with our co-receptors, right, we get a conformational change. So this glycoprotein here, GP41, changes its shape. And what this does is this facilitates fusion, right, step 2 fusion, of the envelope with the host membrane. So if you remember from the beginning, I said that this envelope came from the host, right? When it comes into the cell, it actually puts it back in, right? So it's a cycle. It enters the cell and it fuses into the cell membrane. And once it leaves at the end of the cycle, it's going to take it with it again. All right, so here we bound to our receptors and then our nuclear envelope has fused with our host cell. Then what happens, right? We're in the cytoplasm, and what needs to happen is what we call uncoating. This is the process by which this cone-shaped nucleocapsid is removed, because all the good stuff of HIV, right, all the stuff that facilitates invasion is in here, the nucleic acid core. Sorry, all the stuff that facilitates latency, I should say more correctly. So we need to remove that capsid, the cone-shaped capsid. And once we do, we're left with the nucleic acid core and its viral enzymes. The first one we're going to go through is called reverse transcriptase, okay? Because once our mRNA is in the cytoplasm, reverse transcriptase plays an important role. it converts our mRNA strands into complementary DNA, cDNA. Now, if we think of central dogma of biology, we always think DNA goes to mRNA, which goes to protein. But we call HIV a retrovirus, and the clue's in the name, retro, backwards, going back, right? It has this enzyme, reverse transcriptase, which allows it to go from RNA back towards DNA. It's an interesting virulence factor of HIV that allows it to incorporate into the host genome, okay? So once we have DNA, we then form what we call a PIC, the pre-integration complex, okay? The pre-integration complex is when that complementary DNA associates with a number of proteins, and it forms almost like a vehicle, if you will, that allows transport into the nucleus. Now, I've written question mark size because there's a really interesting controversy, if you will, about the pre-integration complex, because the size of the pre-integration complex is actually larger than the size of the nuclear pores. Why is this an issue? Well, if you think about it, right? If the pre-integration complex needs to get through this hole, right? It needs to get through these holes that are in the nuclear membrane. The only way it's going to get through is if it's smaller and it can fit. But interestingly enough, this is actually not the case. The pre-integration complex is larger than the nuclear envelope, the pores in the nuclear envelope. So it's actually unknown how it fits in and actually gets into the nucleus. All right, so now we're into the nucleus, right? We have our complementary DNA, it's formed the pre-integration complex and it's transported into the nucleus. Then what's going to happen? Well, we need to integrate into the host DNA, okay? This is our integration step. So we have a second viral enzyme, integrase. What this does is it induces 3' processing. So it causes modifications of the newly reverse transcribed complementary DNA and makes some changes to the 3' end. And these changes are necessary to allow HIV viral DNA to incorporate into the host's nucleus, right? So once you process it, it's now ready. And what we can do is we can transfer, in the transfer step, we can actually physically, mechanically transfer that processed DNA into the host genome. And this is the point of no return, right? This is the point where HIV can establish latency because now it's in the host genome. And so every time the host genome, that particular region of the host genome is transcribed, guess what? The HIV variant, the HIV virus and all its associated proteins are also transcribed. And this is what makes HIV quite a persistent infection. It's really hard to get rid of because it's in our DNA. All right, once we've integrated, what happens is just like our normal cell cycle. Back to normal central dogma of biology. We then transcribe an mRNA strand, okay? And then from that transcription, we translate a protein. Now, one thing I want to mention is that transcription is controlled by a protein named TAT. TAT is the trans-activated protein, and it's involved in the activation of RNA polymerase II, right? The key driver of transcription. And what the trans-activated protein does is it makes sure the RNA polymerase stays on to that DNA. Because without TAT, in its absence, RNA polymerase quickly falls off, and it cannot transcribe the whole HIV genome. So the trans-activated protein makes sure the RNA polymerase stays on and transcribes along the whole genome. Okay. Now, another thing too I've noted here is that we have 2 mRNA strands. This is important because once you transcribe and you form 2 strands, one is going to go on to form your protein, will be translated to form your protein, but the other does not. The other, interestingly, stays as mRNA to become the new nucleic acid core of the newly assembled assembling virion, right? HIV is trying to replicate. So it's got one mRNA ready for proteins and one mRNA ready to be the next nucleic acid core. All right. Now, once we've taken that one mRNA and translated it into a protein, what's interesting is that you form a really large polypeptide in HIV. And what this polypeptide is, it's a long chain of all the HIV proteins, all like stuck together end on end, right? So if you think of like a line, it's like protein 1 connected to protein 2, protein 3, protein 4, and so on, okay? And in this state, these proteins are immature and non-functional. They can't do anything, okay? Next, we've got our proteins, we've got our nucleic acid core. Guess what we're going to do? We're going to assemble our virion, right? We're going to traffic towards the host membrane, and we're going to undergo exocytosis. And as I said at the beginning, as it leaves the cell, it takes this nuclear envelope and the key proteins with it from the host cell. Okay? And then finally, the maturation step. So this is where the third viral enzyme protease comes into it. So protease takes that large polypeptide that we spoke about and cleaves it at all the right places. And by cleaving it in the right places, it forms protein 1, distinct from protein 2, distinct from protein 3. And these proteins now cleaved are mature and functional. In its absence, right, in the absence of cleaving, the HIV is no longer actually infectious. Okay, and that's the life cycle. Just really quickly, I'm not going to go into any HIV drugs in this video, but what I wanted to point out is if you look at the stars that I've put, right, what I'm trying to illustrate by these stars is there are a number of steps where we can target pharmacologically to stop HIV infection. And this has been a major driving force of HIV treatment. So if you think about adhesion, right? If we're binding to CXCR4, for example, right? Or CCR5, we can try and form something that's going to block these receptors, right? Maraviroc as an example, yeah? We have reverse transcription. You can try and inhibit the reverse transcriptase enzyme, right? Our nucleoside reverse transcriptase inhibitors, our non-nucleoside reverse transcriptase inhibitors, right? Here, integration. We have another enzyme, integrase. What can we do? We can try and inhibit the enzyme, right? Transcription, the transactivated protein. Let's try and inhibit that, right? When you're cleaving the polypeptide at the end by protease, let's try and inhibit that enzyme, right? So in any infection, it's really important, or in any disease, really, it's really important to try and establish the pathogenesis, because that's what establishes where we can target our drugs, where we can develop drugs to stop the progression of disease. And so that's it for today. I hope that was helpful. We've gone through the life cycle of HIV infection, and really quickly at the end, identified some drug targets from that life cycle.